Okay, good morning everyone. Sorry for the delay. So today is the third lecture for this Introduction to Computational Molecular Biology course. And today we are going to talk about DNA sequencing. So many of you probably knows about DNA sequencing before, but today we're going to review like all the techniques from the first generation to the third generation, and then look at some kind of application that we can do, some molecular assays that we can do with DNA sequencing. Okay, so today's objective is simple. We will talk about DNA sequencing platform, what they are, what are their pros and cons, and then we will look at some of the applications that use them in biology. So just in case any one of you are not familiar with the sequencing techniques, so everything starts with the discovery of DNA polymerization, the fact that in our cells we can duplicate our DNA by having individual nucleotides that are being incorporated in a polymerization reaction, so we can generate new polymer from a template. And usually this can only occur if you have a correctly matched nucleotide like A to T or C to T. And this reaction occurs on the 3' hydroxyl group, so you have to have this in order for the phosphate group from the next nucleotide to form the reaction and then incorporate it into the DNA strand. So in the early days, scientists kind of exploit this fact that if you don't have this hydroxyl group, the polymerization cannot go on. So if you introduce another molecule called DDNTP, so usually you have one hydroxyl group, but in DDNTP, dideoxynucleotide, both the hydroxyl groups are gone, so now you only have hydrogen. So you can imagine that now if you don't have this hydroxyl group, then you cannot incorporate the next nucleotide. So the polymerization has to stop. So we can use this fact to develop a sequencing technique. Some of you might have seen this in the lab before. It's called Sanger sequencing. So this is the first technique that was invented. And we can imagine that if we introduce regular nucleotide except G, like G we use DDGTP. So only G doesn't have the hydroxyl group. So every polymerization reaction that goes on in the sample have to terminate at G, because some of the G's doesn't have the hydroxyl group. So you have a whole lot of different products. Some will incorporate normal G's and then the DDGTP. Some will incorporate 2 G's and then DDGTP. So you can generate all these different products with different lengths. So these are nucleotides with different lengths. So if you run these molecules on the gel, which will be separated by length, the shortest nucleotide, TCG, will flow fastest. So this will be your bottom band. And the next one, TCGG, this will be the second band because it's the second smallest. So on the gel, if you run it, you will see bands corresponding to all these different kind of possible products. And if you repeat this experiment, not with DDGTP, but with DDATP, DDCTP, and DDTTP, you will get 4 lanes of gels. Each lane you get the band that corresponds to the different kind of nucleotide length. And from this you can deduce that so the first lane here is A, C, G, and T by the alphabetical order. So we know that the smallest product is A. So if we assume that the smallest product is just a single nucleotide, so A would be the first sequence. And then the next one is G. So the second product would be AG. And the third product would be AGC. And the fourth product would be AGCC. And the fifth product will be AGCCT. So we can kind of deduce what nucleotide sequence are being produced in this reaction by looking at how their molecular weight kind of stack to each other as a ladder. But this requires you running the experiment 4 times and then run the gel in 4 lanes. But if you label the DDNTP with different fluorescent color, then you can run them all on the same lane, and then you activate the fluorescent, and then you can see the color corresponding to A, C, G, and T. So this is how we kind of read Sanger sequencing. So we make all the possible kind of nucleotide product, and then we run it on the gel. We activate the fluorescent that correspond to different DD-NTP, and we look at the color. If it starts with blue, then that means the first nucleotide is T. If it's followed by pink, then the next nucleotide is C. And if you have received kind of this type of data from the lab before, if you look like this kind of graph on the right, on the bottom right, you see like the signal in 4 colors. And if the signal is high, then you are confident that that product is that nucleotide. But if the signal is low, then the confidence is lower. So this is based on kind of generating all different lengths of the nucleotide. So you can imagine that this technique is very wasteful, right? If you want to sequence a nucleotide of length 100, then you need to generate a length 1, 2, 3, 4, 5, 6, 7, 8, right? So you need to generate a whole lot of nucleotide strands just to sequence like 100 base pairs. So, but this technique is still good if you want to confirm short sequence, like if you design a vector, if you design a primer in the lab, this can give you a confirmation that your design is correct without having to send it to full-scale sequencing. So it's still in use today in the lab. But if we go to a kind of more high throughput, like if we want to screen all mutation in the patient, for example, then we cannot do Sanger sequencing. So this comes to the next generation sequencing. So this is the term that you probably have heard before. It's the second generation. So first generation is Sanger. Next generation are second generation. Back then they did envision that there will be third or fourth generation. So they just call it the next generation. And the easy way to think about the next generation sequencing is that it's very high throughput. So instead of sequencing one sequence at a time, we are going to sequence millions of sequences at a time. And there are many ways to parallel the reaction. You can separate the DNA sequences into different wells. So each well will consist of different DNA. And you can do sequencing in separate wells. And you get the readout from all the wells kind of in parallel. So this is a technique that they use for 454 pyrosequencing. If you use Illumina, which is the dominant technology today, they also create kind of cluster of sequence. And we will see more details about this. But the idea is for the next generation sequencing, we want to do millions of reactions at once, not just one. And one benefit that we get from next generation DNA sequencing is now we are able to sequence both ends of a DNA fragment. Right? With Sanger, it's only one end. Right? You start from 5' to 3'. But sometimes you just need information from short sequence from the 5' and short sequence from the 3'. And you can use that information to map the location of your read without having to know the full sequence. Right? Because imagine that if you have a DNA fragment, right, and you want to know which chromosome does this fragment come from, if you have a forward read, like maybe just 30 nucleotides from the start and 30 nucleotides from the end. Like if you map these two locations, if you map these two sequences, the first might map to, let's say, 3 chromosomes, and the second may map to another also 3 chromosomes. But because we know that this DNA fragment must come from the same chromosome, we can kind of intersect the answer that we get from the forward and from the reverse read, and get the confident answer that, okay, this must come from which chromosome. So this is the benefit of being able to sequence both the start and the end. Okay. And we will kind of talk more about this once we go into transcriptomics and other techniques, because we will use this ability that we can detect the start and the end to be able to distinguish splice junction, to distinguish gene fusion, for example. So the start might come from one exon, and the end might come from a different exon. And that will be evidence that there are splicing between these two exons. Okay. So this is just a preview for now. And to imagine how DNA molecules that we extract from the cells or from the sample were amplified in DNA sequencer, this is a diagram from Illumina technology. So we start with kind of single molecules of DNA. Each DNA, we only have one molecule from the start. And they amplify this by doing a series of PCR reactions inside the flow cell. so that from the start you get only one molecule each from different part of the chromosome. But in the end, you're going to have a cluster of identical DNA forming. So each cluster will originate from one DNA molecules and got amplified into maybe tens or hundred copies. So when you do the sequencing, instead of getting a signal from one polymerization reaction, you get a combined signal from 10 or 100 of reactions. So you get kind of more confident signals, more clearer signals. So this is a kind of more detailed step by step. So these DNA molecules are flexible, so they can induce the DNA molecule to kind of fold onto the flow cell itself and get PCR reaction. Like you can fill in the nucleotide, so that starting from one molecule, now you get two copies, and the two copies will kind of isolate from each other, and then this loops back onto the flow cell and then get copied again. So you can start from one molecule and then get a large number of DNAs in the same vicinity next to each other. So this is how we can kind of amplify the signal from low number of DNA into kind of large number of DNA molecules. So next would be, once we amplify the DNA, we want to know the actual DNA sequence. Right? We need to do the polymerization reaction and need to read out which nucleotide gets incorporated. So there are several ways of doing this. In the early days, people realized that when a nucleotide is incorporated into the polymerization reaction, one of the products is the phosphate, the pyrophosphate group. And this phosphate can react with enzymes like luciferase to create light. It's visible light. So we can use this as a sensor for the reaction. If you see light, then that means the nucleotide gets incorporated. If you don't see light, then there is no incorporation. So we can pass in ATP, DATP, DCTP, DGTP, DTTP one at a time. And if you get light, then that means that nucleotide gets incorporated. So in this case, if the template is a G, right, and you put in a C, then you get a polymerization reaction and you see the light. But if the template is a G, but we put in A instead, right, A wouldn't be incorporated, so you will not see any signal. You will not see any light from the reaction. Another technology, instead of using the enzyme to generate visible light, they look at the change in the pH instead. Because when a phosphate is generated, the pH will change. So you can monitor the change in pH to say whether the nucleotide is incorporated or not. So these two technologies are similar in a way that they sense the product, the pyrophosphate that were generated by the polymerization reaction. So this is some example. So once we put in, let's say G, right, into the reaction, any wells that light up, these will be wells that the template contain a C, because we put DGTP in the reaction and we get light. So every well that we see here will be the wells with a C, and the other well that are in gray will contain a different nucleotide. So we can keep cycling through A, C, G, and T, and then we see different, and then we see light signal from different cells. So we can accumulate that information to reconstruct the full kind of DNA sequence. The problem about these two techniques is when you monitor light or you monitor pH, the signal is not linear. Right? If you get G incorporated twice, you are not guaranteed to see twice as bright for the light or twice as difference on the pH. So when you have repeat of the same nucleotide in your sequence, there can be problem. You don't know whether this signal that's higher than the other, does this correspond to 3Gs or 4Gs? Because the signal can be very similar. So we call this problem homopolymer. So when your template is a homopolymer, homo means the same. So you have a repeat of A, C, 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 G, 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 or T, 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 T. You cannot distinguish them very well with the pyrosequencing and ion torrent technique. This is where Illumina technology solves. So instead of letting the polymerization reaction occur more than once, if you have a C and C, like if you put in a G, then you can have two incorporation, you can have two polymerization reactions. But for Illumina technology, they block the polymerization by putting a molecule tag at the OH site. So these molecules will block the polymerization reaction. Even if you have 3 A's, only 1 T will be incorporated because these molecules block the polymerization. So in Illumina, each round of reaction will involve an additional step where they have to do a enzymatic uncapping of this tag in order for the polymerization to continue. So with Illumina technology, even if you have a homopolymer, 4 A's in a row, you will incorporate 1 T at a time. So this allows the technology to get the sequence readout to be very accurate. There is no skipping 2 A's, 3 A's. It's always one reaction at a time. But you can imagine that this comes as a cost, right? Now you have 2 steps reaction. So you will take longer for the sequencing to run. So compared to the other techniques, Illumina will take longer, but you get more accurate readout. And while the other techniques use change in pH and visible light, Illumina goes back to fluorescent signal like Sanger sequencing. So each nucleotide will be tagged with a different color. So the data from Illumina will be a series of images. So they're going to image the flow cells, and if they see a blue color, then that means this flow cell incorporate a T. If they see a green color, then this position incorporated a G. And if it's orange, then it's a C. And once we align all these images together, and then we focus on a certain position, like we can read out the sequence to be T, G, C, T, A, and C. So this color tells us which nucleotide, and the position on the image tells us that these are from the same DNA molecules from the same location. So these 3 techniques, I'm going quite quickly because I'm pretty sure you're familiar with most of this, but we're going to slow down for the 3rd generation because that might be new to you. So among next generation sequencing techniques, there are pyrosequencing that use the visible light, Illumina that use fluorescent, and Ion Torrent that use the pH. So the trade-off is Illumina is going to take longer to run, but you get more read at the end, and you get more accurate read. But if you prefer shorter runtime, Ion Torrent, which monitor the pH, is the fastest. So if you want to get answer within a day or within the morning, then you can use this technology. But nowadays we will talk about nanopore sequencing, which will be even faster. So essentially, only Illumina might be considered today for use. Okay, and this is a trade-off for every technology. For DNA sequencing, we care about 2 metrics. The first one is the throughput, like how many nucleotides you can read within an hour. But the second metric is how long of a nucleotide that you can read. With Sanger sequencing, even though it's very wasteful, but it can read nucleotide up to 1,000 base pair, which is much longer than what you get with Illumina or other techniques. With Illumina, beyond 300 base pair, you start to get very low quality. So that's the limitation for the technology. It can read a lot of nucleotide. It can read the nucleotide very accurately, but mostly up to only 300 nucleotide, only up to the 300 base pair. And the reason is because imagine that these reactions still require the polymerization, right? So it still requires enzymes, and enzymes activity degrade over time. So it cannot incorporate more than 300 base pair without degrading the specificity. So once you reach 300 or 500 reaction, the enzyme can incorporate C instead of T or A instead of G. So you start to get those problems. And that's the limitation for the next generation technique, the 2nd generation technique. And if you are curious, so this is like the evolution of DNA sequencing. Everyone starts from having short read length and very low throughput, and then different companies improve their product to get longer read and higher throughput. And in this green region, this is a longer read but lower throughput. This is Pyrosequencing and Ion Torrent is here. Illumina focus on shorter read but more throughput, so Illumina are up here. And next we're going to talk about the third generation sequencing, which are all the way here. We're going to get reads that are up to 100,000 base pair or a million base pair. But the throughput is going to be lower than Illumina. This is a trade-off that we have to make. Okay. So for the third generation sequencing, the key feature is long read. It's up to 100,000 and million base pair. And there are two main technologies that we use for this generation. The first one is called smart sequencing, a very clever name, single molecules real-time sequencing. And this kind of capture the essence of third generation. First is single molecules. With the next generation sequencing, the first step that we talked about is the amplification of DNA. They need a lot of DNA to get enough signals to read the nucleotide. But for third generation, you don't have to amplify the DNA. Each DNA fragment that you get can be read, and you will get the nucleotide sequence from that. So there is no amplification bias. The second part is real time. So instead of having to do the reaction in cycles, like incorporating one nucleotide at a time, or having to uncap the molecules, this technology will do the sequencing in real time. You don't have to control anything. So for smart sequencing, the big discovery that they have is they found a DNA polymerase from some organism that I forgot, but it's very durable. So usually DNA polymerase, I told you that for Illumina, it can only goes on for 300 or 500 reactions, but for these polymerase, it can goes on for thousands or 100 or 10,000 reactions. And another technology that they invented, this kind of zero mode waveguide sounds very nerdy. It's a technology where if you want to sequence a single DNA molecules, like you need to make this well, where you parallel the reaction, you need to make this well very small. Otherwise, you have multiple DNA molecules occupying the same well. But when you have the well being very small on the nanometer scale, then you have problem with interference with light signal, with fluorescent signal. So this is like a physics or engineering improvement that allow us to monitor fluorescent signal within a very small space. Okay. And when I tell you that this is not when I tell you that this is real time, for smart sequencing, it's not imaging each cycle, one image per cycle. They are taking a video of the reaction. So you will see signal over time. If there is an incorporation of a nucleotide, you will get fluorescent signal from that color as over time until the reaction is done. Then there is no polymerization, and then there is incorporation of an A, and then no polymerization, and then there is incorporation of a T. So you might think that, okay, instead of getting images, now we got a video. What use can we make from video, right? We just see the same incorporation of C, A, T, G, and G. But the thing is, when we look at the polymerization reaction, if we have a modified amino acid, like methylated amino acid versus regular amino acid, it's structurally different and chemically different. So even though it's the same incorporation of an A, but the incorporation of modified A and regular A will take different amount of time. So with SMRT-seq, they can look at the interval that the polymerase, the enzyme takes between each incorporation to tell whether the nucleotide, the next nucleotide, whether it's modified or it's unmodified. So you can study not only the nucleotide sequence, but you can also detect the nucleotide modification at the same time from this technology. One of the key limitations of third generation technology is the error rate. Even though they found polymerase that can operate on a scale of 1,000 or 10,000 reaction, it's not a very accurate polymerase. So you will get 5 to 15% error rate. So even though the sequence should be, a T should be incorporated, 5 to 10% of the time, the polymerase will accept a G. So it's a mistake. But the good thing is, this polymerase make the mistakes at random. So one way that we can solve this problem is we can sequence the same molecules several times. So imagine if you can read the same DNA molecule 10 times and you make some errors on each read, right? But if you do the kind of consensus voting, right? If you look at the majority would be correct because the errors are random. So in this case, this position, there's one C, but a whole bunch of G. So if you take a voting, you will know that the sequence here should be a G, not a C. So people who invented SMRT-seq have to also invented how to sequence the same nucleotide, the same DNA several times. So what they developed is called a circular consensus sequencing. So they turn their linear DNA into a circular DNA. And with circular DNA, the good thing is you can do polymerization reaction multiple times and you can create multiple copies of the DNA over and over. And they found that if you do this polymerization reaction 10 times, so for each DNA molecules, you circularize it and then you amplify it 10 times. Not amplify into different molecules, but because it's circular, when you amplify it and you unwind it, it will still be one continuous DNA. But you get the same sequence repeated over and over. So when this molecule is read by the SMRT-seq, you will get kind of one single read that's very long, but within that single read, you will have multiple copies of the same sequence. And then you can take the consensus voting for those sequences. And they found that if you do this, you extend the DNA molecules into 10 copies, you can reach the same error rate as the Illumina technology, which is 0.001%. So basically you spend more time creating longer DNA, but because you have a durable polymerase, you can read the whole thing. And then you take the consensus voting, so it's kind of on the software side, you do the consensus voting and then you get the correct DNA sequence. Okay. And with this technology, we can get reads that are up to 10 kilobases, 100 kilobases. So this allows you to study things that were normally unavailable to kind of short read data. And we'll talk about this later. So that's the first technology. The second one that I want to introduce is called Nanopore sequencing. It's called Nanopore because the technology evolved around a protein that acts like a pore. So it has a hole in the middle and it's small, so it's nanopore. So this technology is very interesting because in the future it can be adapted into different things for different assays. The idea for nanopore is that all the technology before were limited by the polymerization reaction. Right? We cannot make it faster, we cannot make it longer, we cannot make it more accurate. So nanopore people thought about DNA sequencing in a different way. So imagine if you can pull DNA strand through some porous protein. As the nucleotide got pulled through the pore, it will block the flow of ion through the pore. And because nucleotide have different structure, you can imagine that different sequence will create different signal of blocking the ion flow. So what they monitor in nanopore is the flow of ion across this protein pore. This is a graph of the signal. It can be low, so it's blocked. It can be high, so the flow is good. It can be low again because it's blocked. It can be high, the flow is good. So this signal must be transformed into nucleotide sequence. Right? So this is very cool because who would know how to kind of transform this? Like with DNA polymerization, we exploit kind of nature that they have a perfect pairing between AT and CG, so we know what happens. But with this information, now it turns into a physical data. Right? What is the structure of nucleotide at that area? How can we turn this into sequences? And the answer is we can't. But what we can do is, now that we have technology that can synthesize DNA, you can generate synthetic DNA sequence with known, you can create synthetic DNA with known sequences. And you can specify A, T, C, C, G, you can synthesize it. And once you synthesize the DNA with known sequence, you can put it through nanopore, and you get the signal. So even though we cannot turn from signal to nucleotide, because we can synthesize any nucleotide that we want, we can generate signals for those nucleotides. And once we have those patterns, like nucleotide and electrical signal, we can train a prediction model that then look at the signal, try to figure out which nucleotide it is. So we can decode it by generating a lot of training data. And this helps from and this kind of machine learning approach, which we will kind of talk about toward the end of the class, help reduce the sequencing error from 14. So this is similar to SMRT-seq, like without the circular consensus sequencing, down to 1 to 5%. It's still not Illumina level. It's not 0.001%, but it's pretty good for many applications. The requirement is you need very powerful computer. Especially you need a graphical processing unit or GPU to run this kind of decoding model. For nanopore, if you want to get accurate read, because nanopore doesn't have the kind of circular amplification process, instead what they introduce is what's called a UMI, or unique molecular identifier. So the idea is before we amplify the DNA, the problem with DNA amplification is once we amplify the DNA, we cannot trace how many original DNA molecules there are. We only know the product after amplification, and that can be biases. Some DNA got amplified more easily than the other. But with unique molecular identifier, and we will see this concept again in the single cell technology lecture, if you can tag each original DNA molecules with a unique nucleotide pattern, when you amplify that DNA molecules, that unique nucleotide pattern will also be amplified. So this is kind of unique to each original DNA molecules. So once you do the sequencing at the end, any reads that have the same tag must originate from the same molecules. So you now know that even though I have 50 reads here, these are all originated from the same DNA. So they must have the same sequence. And once we have this information, we can do a consensus voting again. And if we have 50 reads that have the same UMI, the same tag, so we know that this must come from the same DNA, and so we know that they must have the same sequence. We can look at the voting among these 50 reads, and then we can get the correct DNA sequence at the end. So this is another technique that we can use to correct the error from third generation sequencing. And similarly, because now we look at the electrical signal that were caused by the kind of the structure of DNA strand that blocks the pore. So when you have modified nucleotide, they will have different structure, so they will block the pore differently. So you will get different in signal that can tell the decoding software that, okay, this is a modified A and this is a regular A. So we can look, the software understand from the signal that this is a modified nucleotide, not a normal nucleotide. Okay. So with both technology, even though they come from very different concepts, they achieve the same ability. Both are able to read DNA molecules at single molecules level in real time, and they both are able to identify modification on the nucleotide. For nanopore, you can achieve kind of read length up to megabases. So you can sequence basically the whole bacteria and the whole virus in a single read. So it's very good for those applications as well. And for nanopore, one of the unique characteristics is very portable. If some of you might have seen the actual sequencer before, right? If you imagine Illumina or SMRT-seq, PacBio sequencer, it's going to occupy like this whole table, basically. But for nanopore, if you want to sequence in small scale, it's the size of your USB drive. Maybe USB from 20 years ago, but it's the size of a USB drive. So you can bring this into the field. Like if you go to outside of Bangkok, you go into the wood, you go in, and you get sample from like wildlife. You can do sequencer right there. You don't need to collect the sample and bring it back to the lab. So that means you can do sequencing kind of not only off-site, but you also don't need to invest a lot into the machine. You might need to spend million of baht to buy a Illumina or PacBio sequencer, but each nanopore, the small scale nanopore, I think is to the scale of thousands or 10,000 of baht. So you can kind of start your sequencing project with low kind of overhead costs. And I mentioned earlier that nanopore is very fast. So with Ion Torrent, you get the result in 2 hours, but you get kind of short read, and you have problem with homopolymer. With nanopore, including all the sample preparation, you can get the full sequencing, the long read sequencing, within a day. But if you want to have the answer very fast, the software that decode the signal have the fast and the slow mode. So if you use the fast mode, you can get the result within 6 or 7 hours. So it's a time scale where you can use it in hospital. You can get blood sample or urine sample from the patient, run into a nanopore, decode the data in real time, and then get the recommendation to the doctors within half a day. Another feature that's very cool with nanopore, so with other sequencing techniques, you have no control over the sequencing process. You put in the DNA, and then you let the polymerase kind of do the reaction, and then you get the signal kind of at the end. For nanopore, because the process of sequencing involves the pulling of DNA strand through the pore. Right? So imagine that as you pull the DNA strand through the pore, and the software start to decode the signal, so you start to get the read for the first few nucleotides, right? And if you somehow have a knowledge that, okay, I'm going to, let's say you are studying a clinical sample, like you get a blood sample from the patient, so it's going to be a mix of human DNA and pathogen DNA. And let's say you want to screen for pathogen DNA, so you know that anything that looks like human DNA, you don't even want to sequence. But sometimes, the human DNA will get pulled into the pore. But the cool thing is, the software, once it decodes the DNA, and it gets enough nucleotide sequence, then it can start to match to human genome and know that this is human DNA. It can sense the signal to the pore to, instead of pulling the proteins, the DNA further, it can reject the DNA. So you get an adaptive kind of sequencing process where you can reject DNA that you don't want, and you can continue to sequence DNA that you want. So this is a feature with kind of nanopore technology. Because you can monitor the DNA sequence in real time, you can tell the pore to continue sequencing, or you can tell the pore to reject that protein, that molecule. Sorry. Okay. And I don't have a slide here, but just want to mention if you are interested, because nanopore involves the process of pulling molecules to the pore, right? That molecule doesn't necessarily have to be nucleotide. So people are starting to explore if this technology can be used to pull protein, so that you can sequence the protein, or you can maybe detect a different molecule as well. So it's more flexible than other sequencing technology in this regard. And I think within a few years, you might start to see protein sequencing with nanopore in some shape or form. So you can look out for that. Okay, so with both technology, we can get very long reads. One of the benefits is if you want to study repetitive regions, so these are regions where you have kind of repeat of the same nucleotides kind of over and over. And you might be surprised that as much as 20% of the human genome is like this. So these colors kind of stand for these 4 different nucleotides. There are many regions in human and other organisms where you only have mostly GNC. They call this CGI. There are a whole bunch of C and G bunched together. And you can imagine that if you get a short read, like the scale of my blue bar here, there is no way that you can identify where this comes from. It's all going to look similar, all C and Gs. But if you do third generation sequencing with the long read up to 100 or a million base pair, you can sequence the entire region. So this entire diagram here is one read from a third generation sequencer. So you can study this repeated region in full. So this is one benefit. You can get coverage into the region where previous technology cannot give before. Another benefit is if you want to study haplotype. So the idea of haplotype is we know that mutation in the patient, in any organisms, the mutation sometimes are not independent. You get some mutation that kind of got transferred together to evolution. So for example, this A and A are co-occur together. So in one patient, in one person, he might have or she might have an A and an A here, but another person might have a G and a G. So it's kind of correlated, it's linked together. With Sanger sequencing, because we produce kind of different DNA product, we have no idea. We can tell that there's an A and a G there, but we will never be able to tell whether they occur together. For second generation sequencing, if the mutation occurred near each other within 2 to 300 base pair, then you can tell that, okay, every time I see an A here, I see an A here. But if there is another mutation further away, more than 300, more than 500 base pair, you cannot link this to this earlier mutation, because in the sequencer they will occur on different molecules. But for long read sequencing, you will know that this A and this G occur together as a single haplotype, as a single block, as a single unit. So this is another benefit in kind of genetics that people like from third generation machine. Today, now that we have both kind of next generation which are short read, better throughput, more accurate, and third generation which have longer read, lower throughput, and less accurate. People start to put these two technology together, and this is a strategy that I hope all of you should kind of think about going forward. I think we are beyond the period where we select either short read or long read. Going forward, it's going to be a mix of the two. So imagine that if you study a new organism, let's say you identify a new animal, and you want to recreate the whole genome. If you do only short read, then chances are you're not going to be able to reconstruct the whole genome, because with short read you don't have a full coverage. In some region you cannot assemble the data. With long read, you're probably going to see the entire chromosome, but you're not going to be so sure about the actual DNA sequence. If you combine the best of both worlds, you do long read first, so you get a good idea of the chromosome, like how long it is, the sequence composition of each region, and then you also do short read, and then you map the short read to the long read. So the long read will serve as your scaffold, as your template, and you might not be confident with the nucleotide sequence in certain region, but once you map all the short read into this region, you will get like 10 or 100 copies of DNA from that region. So you will get kind of start to be more confident about the actual nucleotide sequence. So people start to combine kind of these two techniques so that you can get both the complete coverage and also the accurate nucleotide from both technology. So it might be more expensive. Money is always an issue, but in terms of the techniques, this is something that can solve many problems. Okay. So just to summarize, we have today, I think we can kind of only focus on Illumina as a representative for the second generation techniques. So we have Illumina competing with SMRT-seq from PacBio and Nanopore. So in terms of read length, you have a scale of 50 to 300 base pair, all the way to 25 kilobase to 100 kilobase or megabase. In terms of run time, Nanopore is by far the quickest, less than 10 hours, and the traditional polymerization based method is going to take like a week to run. Okay, so you might kind of have a question that, okay, so when should I use kind of each technique? The best way would be to kind of read the paper on the topic that you want and then see which platform they use. But as a general kind of rule of thumb, like if you want to identify low frequency event, like low frequency mutation, you want to sequence with high throughput because you want to detect something that's very rare. So in that case, Illumina would be best because you get many reads from Illumina. You can detect like a 1% mutation from the cancer sample. If you want to quantify gene expression, you already know the genes, like let's say human cells, you know all the genes, you just want to quantify the expression level. Then you also need many reads because you want to quantify it. So in that sense, Illumina is also good. If you do a targeted sequencing, like specific genes or specific region of the genome, because the scope is small, you can focus on the throughput. Right? So in that case, Illumina is also a good choice. If you want to assemble a new genome, like we mentioned before, a mix between short and long read would be the best option, if you have money, of course. If you want to go into the field, or if you want something that's very fast for clinical service, then Nanopore is something that a lot of people can consider because of the unique characteristic of the technology. But you have to deal with the sequencing error that's higher than the other technique. But if you want to be very careful, you want to study structural variant, like the chromosome, one chromosome moves to another chromosome, like those kind of events. Or if you want to copy if the gene copy number change, like you get additional copy of the gene where they are. You want to know those exact details on the structure of the DNA, then you need long read, but also accurate DNA. So in that case, you need the SMRT-seq from PacBio. Nanopore can give you like the roughly what happens on the genome, but if you want to know the actual mutation at the same time, you want to identify whether there are different genes being copied, then you need to be accurate. You need the accuracy on the nucleotide readout. Okay. So with DNA sequencing, like we know that we can sequence DNA, but actually we can do more than just sequence DNA. So for the last section of today's lecture, we're going to talk about kind of molecular assay that we can create with DNA sequencing. But first, I want you to kind of consider that when we think about DNA sequencing, we can be flexible about the scope of sequencing and the depth of sequencing. So this is because when you pay for DNA sequencing, the cost scale based on the base pair that you get. So if you sequence the whole genome, you're going to pay more than if you sequence just the exome or the coding part. And it's going to pay cost more than if you sequence just one gene. In terms of depth, if you sequence the genome 20 times, it's going to cost more than if you sequence the genome 2 times. So you have this option. Do I need the whole data, or do I just need some part of the genome? Do I need 30 read, 30x coverage of the entire genome, or do I just need just 2x or 5x? So this depends on the application. But for example, if you are studying kind of mutation that cause disease, as a first, as a preliminary study, usually you want to focus on protein coding genes. So those will be exons. So you can reduce the cost by not doing the whole genome sequencing, but doing the whole exome sequencing instead. But if you have very specific research question, like you want to study the composition of bacteria, and with the bacteria, we know that we can just sequence its ribosomal RNA or RDRP gene for the virus, and then we can kind of classify its taxonomy. So we don't need to even sequence the whole genome for the pathogens. We just need to sequence one gene, and then we are able to tell which strain it is, how it related to other genomes. In the case of cancer, we know that not all genes contribute to cancer. So in some cancer, people have a good idea of which genes should be monitored. So you, instead of sequencing the whole exome, you might select 100 genes that are very frequent in certain cancer, and you can sequence only those genes. And if you kind of go search in kind of Illumina company, for example, they also start to provide service where instead of sequencing the whole genome or the whole exome, they will give you a set of genes, and then they say that, oh, if you are focusing, if you are interested in immunology or immune cells, you can focus on these 100 genes. If you want to study breast cancer, these are the 100 genes that you can sequence. So that kind of saves you cost as well, because you don't need to sequence the whole thing. Another aspect is the sequencing depth. if you want to identify mutation, you want to get a high depth because you want to make sure that if the mutation rate is 2%, then you need to see, let's say, 100 molecules, and 2% of the molecules will contain the mutation. So you need a lot of reads for that. But if you are studying like a germline mutation, where you expect the person to be either homozygous or heterozygous, right? It's only 50% or 100%. You don't need 100x. You don't need to have 100 molecules, 100 reads to do that. You can get 10 reads, and then you can detect whether about 5 reads are mutant or not. So you don't need to do a whole bunch of sequencing for those situations. In other situations, in cancer, in some cancer, there is a duplication of the whole chromosome. Right? If you want to detect the duplication of an entire chromosome, you don't need to have the read kind of mapping to the entire chromosome. Right? If you see double the amount of DNA for some part of the chromosome, you can probably deduce that the entire chromosome is duplicated without having to have the read kind of mapped to everywhere. So in this situation, people does what's called a ultra low pass kind of sequencing. Instead of having 100 reads all span the entire chromosome, they require only 2 or 3 reads spanning the chromosome, and they are able to tell whether the chromosome is duplicated or is lost just from those amount of information. Okay, so the idea for kind of targeted sequencing is we can enrich the DNA for the one that we want to sequence. The reason why we are able to do exome sequencing for human, for mouse, is because we know all the exons. So once we know all the exons, we can design all the probes that are complementary to the exons, and then we can enrich these exons and then do sequencing. Right? It's not there's no magic, right? If we have DNA in the cells, right? How can we extract just the exon, right? Because they are all DNA. The only reason that we can target exon is because we know the sequence for the exon, and we can design profile. The reason why we can sequence RDRP gene or 16S rRNA is because we know the sequence and we can design profile. So the key for targeted sequencing is, do we know the sequence? Can we design the probe? If we can, then we can do the targeted sequencing. So at this point, then you might be curious, then how can we do whole genome sequencing? Right? Because the genome, we don't have any pattern. And how can we kind of amplify the whole genome? And the idea is, instead of making a probe that's kind of targeted specific for the exon or for the gene, for whole genome sequencing, we generate random primers. So we generate almost all possibility of nucleotide. So whichever sequence, whichever region of the chromosome, there's going to be a probe that is very similar to it. So we can amplify the whole genome. So it's kind of not targeted. You generate a bunch of random primer. It's going to amplify everything in the sample. Okay. So if you're curious, when we say that we can study bacteria or virus or fungi by studying the ribosomal RNA, the reason is because on ribosomal RNA, there are certain regions that are very conserved. So in whichever bacteria, it's going to look pretty much the same, this blue region. And then there are regions that are variable. So in one bacteria, the sequence will be one thing. In a different bacteria, the sequence will be different. So if you design a primer on the blue region, on the conserved region, you can sequence the variable region. And there are many different variable regions that you can target. And this will tell us, once you get the kind of sequence for this variable region, you can classify which phylum, which genus, which species does the bacteria came from. So kind of we rely a lot on biology when people came up with these kind of assays. So let's say you want to study transcription factor, like we want to know, does it bind near our gene and then activate our gene. So when transcription factor, which is protein, bind to the DNA, we can chemically induce a cross-link between the transcription factor and the DNA, so it will remain bound together. Then we can design the antibody for the protein, for the transcription factor. So we can pull down the transcription factor, which will also pull the DNA strand along with it. Then we can extract the DNA and do sequencing. We're going to talk more in details about these techniques in later lecture, but for now, I just want to kind of show you the fact that DNA sequencing is the final part that can be used for many different kind of assays. Let's say you want to study kind of histone or nucleosome, which are the protein that the DNA were bound onto. These are modified, so we can design antibody that target kind of histone methylation of the histone, acetylation of histone, and then we can pull down the DNA that are attached to that nucleosome molecules, and we can do sequencing. So at the end, we are doing a kind of targeted sequencing, but instead of targeting with a primer, we are targeting by the antibody that binds to the protein that targets certain DNA. So it's very convoluted, right? We target the protein that targets certain DNA so that we know which DNA pattern is recognized by that protein or by that nucleosome. So another cool technique, this one is less well known than ChIP-seq, is called DAM-ID, so it's DNA adenine methyltransferase identification. The idea is if you have an enzyme that can modify certain pattern on the DNA, if you can bring that enzyme onto the DNA, it will make the modification. So you can attach this enzyme to a protein of your interest. If that protein can then bind to the DNA, it will also bring that enzyme to the DNA. So in this case, it brings the adenine methyltransferase to the chromosome. The protein that you are interested in can bind to DNA, to chromosome, and it brings this enzyme together with it. And anywhere that it binds to the chromosome, it will modify, it will methylate the DNA next to that location. So then we can sequence the DNA and then detect for the methylation. And anywhere that we detect methylation is where the protein binds. So we can use we can change the adenine methyltransferase to different enzymes, and the principle will work the same. If you have an enzyme that can modify DNA, you attach this to a protein of interest. That protein, when it was recruited to the DNA, it will bring the enzyme, and you do see the modification. And when you sequence and see the modification, it will be specific on the chromosomes where that protein recognized. So it's similar to ChIP-seq. Like for ChIP-seq, you can detect where the transcription factor binds to the chromosome or where the histone is modified. For DAM-ID, you can study any protein that also binds to the chromosome, and you can look at the pattern of methylation that occur afterward. Okay. Another technique that's also used a lot is called bisulfite sequencing. So in this case, we want to identify cytosine that's methylated. So this is important for gene regulation. If you have methylation, it can change the transcription of a gene. So we want to know in the genome, in the chromosome, whether the promoter or the gene body is it methylated or unmethylated. But in order to sequence the methylation, let's say we don't have the third generation sequencing, we use Illumina. What people does is they perform a reaction called bisulfite conversion. So bisulfite conversion will attack unmodified cytosine, and it will turn into a uracil, which is read as a T. So U on the RNA is read as T. So when we do sequencing with and without bisulfite conversion, so without bisulfite conversion, all the C will be read as C, as it should be. But with bisulfite conversion, any unmodified C will be read as a T. So you can do sequencing on these two conditions, and then you can compare the result, and then you can tell that any location that remain a C, these are methylated. Any location that switch between a C and a T, this is unmethylated. So we can combine chemical reaction together with sequencing to identify methylation of the C. Okay. Another series of techniques, this is to study kind of whether the chromosome is kind of open or closed, euchromatin or heterochromatin. We're going to talk more about this in later lectures about chromatin conformation. But for now, I want you to appreciate the fact that we have enzymes in nature that can digest open chromatin. Like if accessible, the enzyme can attack it. So we can generate DNA fragment from the open region of the chromosome, and then we can select these fragments to sequence. So with one enzyme, you can specifically target open DNA. With another enzyme, which is more sensitive, it can cut this small DNA this small opening as well. You can use this enzyme to target closed DNA, because all the open DNA will be degraded, fully degraded. All that's left is DNA that's bound to the nucleosome. So with different enzymes, you can target different regions on the chromosome, and this tells you different information. If you do this DNA reaction, then the read that you get will map to open DNA. If you do MNS reaction, the reads that you get will map to closed DNA. But if you do ATAC sequencing, you will get a combination of both. So let's see in more detail. So DNase can cut open chromatin, open DNA basically, because the enzyme has to be able to access that DNA in order to cut it. But the enzyme is large, so DNase is kind of large, so it cannot access this small region. It cannot cut DNA kind of in between nucleosomes. So you will get DNA product from the open chromatin. But for MNS, this is another enzyme found in bacteria, Staphylococcus aureus. So this enzyme is smaller, so it can cut individual nucleosome into pieces. If you use a high concentration of this enzyme, you can cut every open DNA, and everything that survives will be DNA that are bound to the nucleosome. So you can study nucleosome-bound DNA using this enzyme specifically. With ATAC sequencing, it's ATAC is from assay for transposase accessible chromatin. So it uses transposase, which is also enzyme that can insert itself into the chromosome. In this case, the enzyme transposase is connected to a sequencing adapter. So if the transposase can access the DNA where it's open and insert itself into the DNA, it will also insert sequencing adapter. So imagine that if you extract the DNA and put it through a sequencing machine, it will only sequence where the transposase can reach, because the other location will not have the sequencing adapter. So no sequencing will be happening, only where the transposase can integrate it into. So these techniques can sequence open chromatin, open DNA, but also sometimes where you only have few nucleosomes, so the transposase can still squeeze itself into the DNA. So these techniques can allow you to distinguish between open chromatin and closed chromatin. And this is important because in later lecture we found that open chromatin is associated with active DNA, active gene expression, and it's important for other regulation as well. Another interesting technique is called chromatin conformation capture. So we will learn in later lectures that we used to think about DNA or the genome as just kind of one-dimensional, like A, T, C, G, 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 A, and just one-dimensional. But because DNA is also a polymer, so it must have some 3D structure. And if you imagine, like we have in human, we have 3 billion nucleotides on the genome, and imagine how small the nucleus is. You have to fold polymer that have 3 billion nucleotides into the nucleus. If the folding is not well organized, we're going to have problem for sure. Imagine if you have, okay, I think most of you have like a AirPod nowadays, but if you imagine back then when you have earphone with a wire, and if you kind of couple it up and put it in your pocket, if you take it out, it's going to form mini loops. So it's the same for DNA. If the DNA doesn't fold properly, it's going to form mini knots, mini loops inside the nucleus. And when you have the cell division, where you need to duplicate the DNA and separate it into two daughter cells, you're going to have problem because DNA going to be stuck with each other. So people start to wonder for a while how DNA can fold so nicely that every time we want cell division, all 23 chromosomes can separate, duplicate, and then goes into different cells with no problem, no delay. So one technique that people use is called called complete conformation capture. The idea is when two parts of chromosome comes together in three-dimensional space, that comes together in 3D. If we introduce a cross-link, so a chemical molecule that lock this into place, then we digest the DNA, and then we induce them to form a loop, ligate them into a loop, and we cut them again. We're going to generate this kind of product, a hybrid product, where one end of the DNA comes from one chromosome, the other end of the fragment comes from a different chromosome. But these two sections of the chromosome appear to be close in 3D. So imagine then if we do pair end sequencing, so we read like 50 nucleotides from one end and then 50 nucleotides from the other end. This 50 nucleotide is going to map to one chromosome. This 50 nucleotide is going to map to another chromosome. Usually that's bad. Usually that's not what you want. Right? If you sequence the DNA, you expect the forward read and the reverse read to come from the same location. Right? But in this case, because we are studying 3D interaction, 3D interaction can come from far away in the genome. So we can use the fact that the forward read and the reverse read map to different chromosomes as evidence that these two chromosomes come together in 3 dimensions. So later on, we're going to study about how these techniques allow us to create this kind of model, where we can estimate where all the 3 different nucleotides are in our nucleus. Okay. So this is another application of DNA sequencing that allows us to study something pretty cool. Okay. And for the last technique, so some of you probably learned about secondary structure for the RNA. So RNA, because it's also nucleotide, they can hybridize in certain parts and form this kind of hairpin structure. So people also want to study this hairpin structure. It's very difficult to do this with a physics method. Like you cannot crystallize RNA easily, so you cannot do what you did for protein. So for RNA secondary structure, people have to be creative. So one trick that I think is quite cool in this paper is that we have RNase, so enzyme that can digest RNA. One is specific to hybridized RNA, so double-stranded RNA. One is specific to single-stranded RNA. So imagine if you introduce one enzyme that's specific to double-stranded RNA into the sample, okay, it will only cut where there is a pairing. So once the enzyme is cut, you get the DNA, you get the RNA, sorry, you sequence it. Okay. The start of the read is going to correspond to the cut site, because once you cut and you unfold it, one end is going to correspond to the wherever the enzyme cut, wherever the enzyme was cut. In another sample, if you introduce the enzyme that's specific to single-stranded RNA, and then you do the sequencing on that part, okay, every read that you get, the start is going to correspond to wherever is single-stranded. So when you compare the result from both sequencing runs, you have this kind of patterns where if this RNA is double-stranded, you're going to get more read from the first experiment. If this part of the RNA is single-stranded, you're going to get more read from the second experiment. So we can use sequencing with kind of the right enzyme to understand that, okay, this DNA, this RNA form hairpin loop. This RNA is single-stranded. This RNA form a loop. This form another loop. This is single-stranded. And you can use this information together with other techniques to reconstruct this secondary structure. So it's very cool how we can combine the knowledge from biology, from chemistry, and then use sequencing as the assay to detect where these kind of reactions occur. So that's it for today. So hopefully, if you have any question, I will remain here, but otherwise we are done. Let me stop the recording.